Hello everyone. So in this session we will discuss the TCA cycle or tricarboxylic acid cycle, or which is also known as citric acid cycle, and also we will see fatty acid catabolism. So before that, I would like to mention that TCA cycle is uh, is an example of carbohydrate metabolism, and fatty acid catabolism when a fatty acid is broken down or oxidized. Right. So we will see two metabolism process for two different biomolecules. One is for carbohydrate, another one is for fatty acid. So in this context, I would like to inform you that in the last class we started the metabolism process of different biomolecules. So we started with the carbohydrate metabolism. So we discussed the glycolysis, gluconeogenesis, pentose phosphate pathway, and then also we discuss about their regulation of the carbohydrate metabolism processes. So, after discussing about the biomolecules, carbohydrate, protein, lipids, nucleotide, nucleic acid, then we are now covering the metabolism process. So in this session, the first TCA cycle will be described, which is a carbohydrate metabolism, and next is fatty acid catabolism will also be discussed. So we will first start with the end product of the glycolysis. If you remember from our last session where we discuss about glycolysis, then we have seen that the end product of the glycolysis is pyruvate. So per one molecule of glucose, you can get two molecules of pyruvate that we understood in the last session. Now, and we also saw some possible fates of pyruvate. One thing is, if the condition is aerobic, then pyruvate or pyruvic acid can either go for TCA cycle, right, which we will discuss today, which will eventually produce CO2 and ATP, or the pyruvate can be used as a precursor for gluconeogenesis, so that the cell can make glucose from pyruvate by the process of gluconeogenesis. So if blood glucose level is low, then gluconeogenesis pathway will be activated. If blood glucose level is high, then the glycolysis pathway will be activated. The TCA cycle pathway will be activated. And if the condition is anaerobic, then in the absence of the oxygen molecule, the cell will use pyruvate to make uh, lactic acid, right? Yeah. So where the basically the pyruvate molecule will be reduced to lactic acid, where NADH will be used as a electron acceptor, which will be converted to NAD plus, right? And in yeast, we also saw in the last session that in yeast, pyruvate can be converted into first acetaldehyde and then to ethanol, right? When the condition is anaerobic. So now we will discuss the fate of pyruvate when the blood glucose level is high, and the pyruvate will be used as a precursor for TCA cycle, right? So till now, the entire as we know that glycolysis process, that is that process is occurring in the cytosol, right. So in the cytosol, from one molecule of glucose, the cell is getting two molecules of pyruvate. Now TCA cycle, if cell has to participate in TCA cycle, then TCA cycle enzymes are in the mitochondria. So the pyruvate molecule, if it wants to enter into the TCA cycle, then it has to enter it into the mitochondria. So how it will enter, that we will see. So the first process is for entering into the TCA cycle is pyruvate will be oxidized to form acetyl CoA and CO2. So this is the pyruvate molecule which we saw in the last session, CHCCO COO minus. So till now it is in the cytosol, right? cytosol, CYT. Okay. Right. So in the cytosol, what is happening? This pyruvate will be converted into acetyl CoA by and this reaction is followed by decarboxylation reaction, and this acid and uh, yeah and this pyruvate molecule will be oxidized. So how? First, this CH3 C11O C00 minus, this CO2 molecule will be removed, right? That's why it is a decarboxylation reaction. So the cell will be left with CH3 C11O, and coenzyme, it will yeah. So this coenzyme will come, right? Coenzyme will come and it will bind to the electron deficient carbon molecule of the CO group, right? This is the carbon which is electron deficient. Here this sulfur group of coenzyme A, which is electronegative, so it will donate electron to this positively charged carbon, so that it will make a bond CH3COS CoA, that is acetyl CoA, right? And it will remove one molecule of Ca2 here. So one molecule of pyruvate will remove one molecule of CO2. So here this is the structure of the coenzyme, right, which is CoA SH is written. So it is composed of it is composed of a modified ADP molecule, right? You can see the nitrogen base is adenine. This is a ribose phosphate, right, which is phosphorylated at the 3' position, right? So that's why it is a modified ADP, and in the C5 position there is two phosphate group, right? So that's why it is ADP, and along with the ADP there is a conjugation of pantothenic acid, right? Right? Pantothenic acid, and then pantothenic acid is conjugated with beta markup to ethylamine, right? So in this beta markup to ethylamine group there is a SH or thiol group, right? If it was OH then it would be all group, but if the O is replaced with S then it will be a thiol group. So now this thiol group has S, and S is sulfur is electronegative, so it will have lone pair of electron, so it will make a nucleophilic attack to this electron deficient carbon here, right, upon removal of the CO2. So now this sulfur atom will make a nucleophilic attack here, so it will make a bond with this electron deficient carbon, which will form acetyl CoA. And the delta G for this reaction is minus 33.4 kilojoule per mole, which is very high. That means this reaction is irreversible, right? Only this reaction is possible for this particular reaction, right? So that's how the pyruvate can be converted to acetyl CoA in the cytosol. So this so this reaction is catalyzed by pyruvate dehydrogenase complex, which is a complex which contains three enzymes E1, E2 and E3 that we will see in the subsequent slide, and it uses Na plus as a cofactor, right, and the electron which is released from this molecule, that will be accepted by the Na plus, so that it will become NADH, right, and it will use TPP or thiamine pyrophosphate as the cofactor, right. So now we will see what is this pyruvate dehydrogenase complex enzyme which converted this pyruvate to acetyl CoA. So this is the oxidative decarboxylation of pyruvate, because there is oxidation, right? It is releasing electron, it is oxidation, and there is a removal of the CO2 group. That means it is decarboxylation also. So that's why this reaction is called as oxidative decarboxylation. Removal of CO2 group along with the oxidation, right? So how does it happen? So pyruvate dehydrogenase is combined of three enzymes. One is pyruvate. So the first subunit is pyruvate dehydrogenase, which is E1. Second one is dihydrolipoyl transacetylase, which is E2, and third one is dihydrolipoyl dehydrogenase, E3. So each of the subunit has individual roles. So first, this pyruvate will come to the vicinity of this E1 or pyruvate dehydrogenase. So it will release this CO2 molecule from the pyruvate, right, by using this TPP as a cofactor. And so if it will lose the CO2 molecule, then it will become CH3 uh, CHOH, and then this electron deficient carbon will be attached with the TPP, right, thiamine pyrophosphate, which is a cofactor. Okay. So after that, that is the job of the pyruvate dehydrogenase, which will uh, catalyze this decarboxylation reaction where the CO2 group is eliminated, and it will make this reaction intermediate, which is also called hydroxyethyl TPP. Right now, the E2 subunit will come, which is dihydrolipoyl transacetylase. So it will use the oxidized lipolysine as the cofactor. So now this TPP CHOHCH3, which is hydroxyethyl TPP, that will react with oxidized lipolysine. So as a result, this oxidized lipolysine will be reduced. So this disulfide bond will be broken, and it will be converted into this S will take one hydrogen to make this SH, right? So it is reduced, right? And then it will release the TPP here, and the CH3CH, right? So the CH3CHOH, so this portion will be oxidized. So from alcohol it will be ketone, so it will be CH3CO, right? And it will be attached to one of this sulfur group of lipolysine, right? So this portion will come here, right, which after getting oxidized, this portion CHOH will become Cu double bond O. The CH3 C double bond O upon oxidation will come and be attached to here, right? So this molecule will be oxidized, so this molecule will release electron, right? And then this oxidized lipolysine will receive the electron to break this disulfide bond, right? So that this sulfur will become SH, and this sulfur will bind with this CH3CO group, right, which is acetyl group. So this is called as acyl lipolysine. Now this acyl, in the step 3, acyl lipolysine will react with coenzyme A, right? So when this will react, then this will be converted into reduced lipolysine, because this portion will be attached to the coenzyme, right? So then it will form acetyl CoA. This portion is attached to the coenzyme A to form acetyl CoA, and this acyl lipolysine will be reduced completely, and so that this sulfur will have a SH group, right? So this will produce reduced lipolysine and acetyl CoA. Now in the fourth step, FAD will come. FAD will will be reduced. It will accept electron from this reduced lipolysine molecule. So FT will be reduced to be converted into FADH2, and this reduced lipolysine will be oxidized. So it will again make this disulfide bond, right? Because it will lose hydrogen. So it will become oxidized lipolysine, right? Now after that, NAD plus will come. NAD plus will receive one electron from FADH2, so that FADH2 will be oxidized to FAD, and NAD plus will be reduced to NADH. Right? This is the fifth step which is carried out. Right? So this fourth and fifth stage of fourth step will oxidize the lipolysine, right? And the fifth step will oxidize the FADH2, right? So these two reactions are carried out by dihydrolipolysyl dehydrogenase, which is E3, right? And this transfer of this acetyl group to the coenzyme A, that part is carried out by E2 or dihydrolipoyl transacetylase. It will transfer this acetyl group to coenzyme to make acetyl CoA, which will completely reduce the lipolysine, right? Right. So this step 3 will be carried out by the E2. Right? So at the end of the day, at the end of the step 5, it it will be, this FADH2 will be oxidized and NAD plus will be reduced to form NADH. So now again after the step 5, this pyruvate dehydrogenase complex will be again ready to for another round of this oxidative decarboxylation of pyruvate. So at the end, by this enzymatic complex PDC or pyruvate dehydrogenase complex, pyruvate will be converted into acetyl CoA. Right. So now, why there is a need of conversion of the pyruvate to acetyl CoA by this PDC? Because pyruvate cannot enter into the mitochondria. So in order to enter into the mitochondria, the pyruvate has to be converted into acetyl CoA. Right? So that's why in cytosol only, pyruvate will be converted into acetyl CoA. So now acetyl CoA can enter into the mitochondria, and we will see by which process acetyl CoA will enter into the mitochondria. So once it gets entered, then it will be used as a precursor for citric acid cycle or TCA cycle. So this is a brief chart of TCA cycle where the first step, citrate synthesis, will take one oxaloacetate and one acetyl CoA molecule to make this citrate molecule. Then there will be a isomerization reaction by aconitase, which will convert citrate to its isomer isocitrate, right? And then there will be oxidative decarboxylation where this CO2 group will be removed. This CO2 group of isocitrate will be removed, right? So here you can see CO2 group has been removed, and this OH group will be oxidized to ketone group, C double bond group. So that's why it is oxidative decarboxylation. So it will release one CO2 and it will oxidize this bond. So it will become C double O minus CH2, CH2, C double O, C double O minus, which is alpha keto glutarate, right? Alpha keto, why? Because at the alpha position from the CO group, there is a ketone group. So that's why it is alpha keto glutarate. After that, coenzyme A will come, and coenzyme A, and this reaction is carried out by the isocitrate dehydrogenase. So from now this alpha keto glutarate will combine with coenzyme SH, right? So that it will remove another molecule of CO2, right? This CO2 molecule will be removed. So another molecule of CO2 will be removed. So this electron deficient carbon will bind to the coenzyme ASH, because as we discussed, coenzyme ASH has this beta markup to ethylamine group, which has a negative electronegative sulfur molecule is there, which is a reactive group, right? That will make an nucleophilic attack to this electron deficient C double bond O, right? Carbon molecule, carbon atom. So yeah, so as a result, at this carbon atom, the coenzyme A will be attached, right? And it will remove one molecule of CO2. So it will become succinyl CoA, right? Then GDP will come, and GDP will be converted into GTP, right? And this coenzyme A will be removed, right? So as a result, the molecule will be CH2 C double O minus CH2, and this coenzyme A will be removed, so it will be C double bond O minus, right? So this GDP will take one inorganic phosphate to make GTP, which is a equivalent of ATP. So this is the first ATP generation step or GTP generation step for the TCA cycle, right? So yeah, so that's why succinyl CoA will be converted into succinate. So now you can ask me that this GDP plus PI to GTP this reaction is not thermodynamically feasible, right? Because GTP is less stable. So that's why from GDP to GDP formation is not spontaneous. But still this reaction is happening. Why? Because succinyl CoA, it is very unstable. You can see because of this C double bond O SCOA group, and there is a negative charge. So that's why this is a very unstable molecule. That's why as it is converted into a very stable molecule, succinate, when it is losing this coenzyme A, right? So after losing, it will become succinate, which is a very stable molecule. So that's why this reaction will be driven into this direction, so that GDP can be converted into GTP by using this uh, delta G difference between succinyl-CoA and succinate. Succinyl-CoA, it is getting converted into a very stable molecule, succinate. That's why this GDP to GTP conversion is happening. Right? This is called substrate level phosphorylation. Right? And when these metabolism pathways are producing NADH or FADH2, they are oxidative phosphorylation. So in the next step, the succinate will be oxidized. No, so from the succinate, one molecule of water will be, two molecules of hydrogen will be removed. That's why it is dehydrogenation reaction. So that's why by this reaction, H2, two hydrogen molecules will be removed, atoms will be removed. So from here one hydrogen and from here another hydrogen will be removed. So there will be a double bond between these two carbons. So C double minus CH double bond CH and then CO minus. So the succinate after this dehydrogenase reaction by succinate dehydrogenase, it will become fumarate. Right? So this is also a oxidation reaction. Right? And here FAD plus will be used as electron acceptor. FAD plus will accept the electron, right, to form the FADH2, right, and these electrons are released by the succinate so that it can be oxidized to be converted into fumarate. And this fumarate, in the next step, the fumarase will add a water to fumarate, so there will be a OH group here and H group here, so it will become C double O minus CHOH, CH2 and C double O minus. So this is malate, right? So now this malate will be oxidized by malate dehydrogenase, which is again a dehydrogenation reaction. So here this CHOH group will be converted. So these two hydrogen atoms will be released, right? So that this carbon will be oxidized from alcohol to ketone, right? So it will become C double bond O, right? So the oxidation. So it will become C double O minus C double bond O CH2 C double O minus, right? Which is oxaloacetate, right? Then again another round of citric acid cycle will begin, where it will combine with acetyl CoA to make this. This is 3 carbon. Sorry, this is 4 carbon and this is 2 carbon. So when they will combined by the citrate synthase, so it will become citrate, which is a 6 carbon molecule, right? So again the same cyclic process will be happening. So in this 3 reaction step, when the isocitrate will be oxidized to form alpha ketoglutarate, where this OH group will be oxidized to C double bond O. So now in this case NAD plus will receive one electron to become NADH. Here also again another dehydrogenase reaction, where this alpha ketoglutarate, this CO2 group will be removed, and so this CO2 group will be removed and it will be attached to with the coenzyme, right? So in this case also the this will release electron which is accepted by the NADH to form NADH, and here in this reaction where the malate will be oxidized to oxaloacetate by, which is also a dehydrogenase reaction, where these 2 hydrogen will be removed, so it will become C double bond O C double bond O here, CH2 C double bond O oxaloacetate. So here also electron will be removed, that will be accepted by NAD plus to form NADH, and another oxidation reaction here, where succinate will be converted into fumarate by losing these 2 hydrogen, right? So that there will be a double bond. So in this oxidation reaction also the released electron will be accepted by FAD plus to form FADH2. So this NADH and FADH2, those molecules can also actually produce ATP by transferring the electron to the electron transport chain, right? So and because of this electron flow in the electron transport chain, the cell will produce ATP by an enzyme called ATP synthase that we will discuss when we will study oxidative phosphorylation. So this ATP generation is called as oxidative phosphorylation through NADH and FADH2. But when we saw in the glycolysis, there was formation of two molecules of ATP in the pair phase, right? So those, and here also from GDP you are getting GTP, which is equivalent of ATP. So this is also substrate level phosphorylation, which is direct phosphorylation, right? So this is the brief of a TCA cycle we talk. So okay, so you can ask me if you have any question regarding this. So after that we will individually discuss the steps of TCA cycle. First, as we know that oxaloacetate will combine with acetyl-CoA to make citrate. So here acetyl-CoA will interact with oxaloacetate. So this reaction is catabolized by citrate synthase, where this coenzyme A will be removed. So it will combine and it will make a citrate molecule. That delta G is very highly negative, so it is a irreversible reaction. Then this citrate can be isomerized by this enzymes aconitase to form isocitrate, right? Just this H and OH will come here and H will go there. That is isocitrate, right? Delta G is positive, so that means the reaction is reversible, right? That's why two arrows are there. From isocitrate, it will take NAD plus to become right. So this is oxidative decarboxylation reaction, where this CO2 of so this C double bond O of which is mentioned here at the C3 molecule, this CO2 will be removed, right? So this CO2 will be removed, right? So that's why it is a decarboxylation reaction, and NAD plus will receive an electron to be reduced to NADH, right? So finally it will become alpha ketoglutarate, where this carbon, this hydroxyl group will be oxidized to ketone group, C double bond. So oxidation here, right, and decarboxylation here. So this CO2 group will be removed, right, and this OH group will be oxidized to form C double bond O, right. So that's why it is alpha ketoglutarate oxidative decarboxylation, right. That is carried out by isocitrate dehydrogenase, where NAD plus will take electron to be reduced to NADH, right. So it will, this step will release one CO2 molecules and one NADH molecule. And the product is alpha ketoglutarate. Next, alpha ketoglutarate will be oxidized again, right, which is again an oxidative decarboxylation reaction, right. So where this CO2 molecule of alpha ketoglutarate will be removed by this decarboxylation reaction, right. So it will become succinyl CoA, and this coenzyme A, it will bind to this electron deficient carbon, right, C double bond. Why? Because as you know that coenzyme A has a negative, electronegative group, which is SH group, which will donate, sulfur is negative, so it will donate its lone pair of electron to electron deficient carbon. For example, here the electron deficient carbon is this carbon, right. So here this coenzyme S will make a nucleophilic attack so that it will be attached to here, right, to make succinyl CoA, and it will remove this CO2, right. This CO2 is removed, so it will become C double bond O, CH2, CH2, C double bond O, and coenzyme AS, right. So that is succinyl CoA, which will remove one CO2 molecule, and the electron which is released, that will be accepted by NAD plus to form NADH, right. So that's why it is oxidation as well as decarboxylation, right. So that's why it is oxidative decarboxylation, and it will also remove one CO2, produce one CO2, and it will produce one NADH molecule. So this reaction is carried out by alpha ketoglutarate dehydrogenase complex. After that, succinyl CoA, right? This is your succinyl CoA. Succinyl CoA. So now succinyl CoA will remove this coenzyme A group, right? So that it will become succinate, right? C double bond O, CH2 and C double bond O minus, CO minus, right? So that is succinate, right? So it will remove this coenzyme A molecule to become succinate, and this, as I told you earlier also, that it will produce one GTP from GTP. This reaction is highly uh, not feasible, right? Thermodynamically not feasible the reaction, but still it is happening here because here succinyl CoA is a very unstable molecule. So now it is becoming a succinate, which is a very stable molecule, right? So that's why as this reaction in this direction, it is forming one stable molecule, right? So that's why there is a energy difference which is used by this GDP and PI so that they can make GTP, right? So that's why overall reaction delta G of this overall reaction is minus 2.9. That means this minus sign indicates that this forward reaction is possible, right? But although the backward reaction is also possible, it is not irreversible, it is a reversible reaction, but forward reaction is possible, right? So again the reason is succinate is very stable molecule, succinyl CoA is a very unstable molecule. So as in this direction it will forming a stable molecule. So this energy difference is used by this GDP and PI so that it can make GDP, right? So this reaction is forming one coenzyme A and one molecule of GTP, which is substrate level phosphorylation, right? Then this succinate will be oxidized to fumarate, right, by using the succ, and this enzyme is succinyl CoA synthetase. The next succinate will be oxidized to fumarate by succinate dehydrogenase. So this uh, two H2 molecules will be removed, right? So that it will be C double bond O. So these two H atoms will be removed. So it will be C double bond O, CH double bond CH C double bond O minus, right? These two H will be removed. That's why it is a dehydrogenation reaction, that is a oxidation, right? And FAD plus will receive those electrons which is released by succinate, and it will become NADH2, right? So this molecule, this reaction step is producing fumarate and one molecule of FADH2. Again, this FADH2 will it is example of oxidative phosphorylation, right? So it can donate this electron. This FADH2 will donate this electron to the electron transfer chain to make ATP by ATP synthase, right? This is oxidative. This is oxidative phosphorylation. So now succinate is converted into fumarate. Now this fumarate will be converted into malate by fumarase enzyme. So this fumarase enzyme will add one molecule of water, so that OH, one H here and one OH to this carbon, so that it will become C double bond minus CH2, then CHOH, and then C double bond minus, which is malate, which is a example of addition of water, hydration, right? So now this malate will be oxidized to oxaloacetate by malate dehydrogenase. So this malate, as we talked about this in last class also in the gluconeogenesis, that malate can be converted into oxaloacetate inside the mitochondria by mitochondrial isoform of malate dehydrogenase. So how this reaction is happening? This in this carbon, this CHOH group, this will be reduced. So these two hydrogen molecule will be removed, so that it will be reduced to, so that this alcohol group will be so actually here two hydrogen atoms will be released, right? So it is a dehydrogenation reaction, and as a result of this, it is an example of oxidation. So this OH group will be oxidized to ketone group, from OH to C double bond. So that's why this carbon molecule will be oxidized here. So that's why it is an oxidation reaction. So it will become C double bond minus C double bond O CH2 C double bond minus, which is oxaloacetate. And here also the electrons, because of this oxidation of malate, the electrons will be accepted by NAD plus to make NADH, right? So that's why it is oxidation reaction, and it will form one oxaloacetate, right? Then again it will cycle in a cyclic manner. Oxaloacetate will again bind to acetyl CoA to form the citrate and all, right? So after one cycle, uh, pyruvate and acetyl CoA and from acetyl CoA to oxaloacetate in this one cycle you can produce. So if you see the balance sheet of this, uh, yeah. So here you can see that if we see a summary of the product for 1 ton of citric acid cycle, then you can see that acetyl CoA will combine with oxaloacetate to citrate, from citrate to isocitrate. Then from isocitrate it will make alpha ketobutyrate, which is the example of oxidation, oxidative decarboxylation. So it will remove one CO2 and it will be oxidized. So NAD plus will be converted into NADH. Again, alpha ketobutyrate will undergo oxidative decarboxylation, so it will remove one CO2, and due to oxidation it will reduce NAD plus to form NADH, right? So it will make succinyl CoA. Then succinyl CoA will remove this coenzyme A to form succinate, and this energy will be used to form GTP from GTP, right? So in this step, the citric acid cycle is producing one GTP, and the succinate will be oxidized to fumarate by succinate dehydrogenase. So here, due to this oxidation, the FAD plus will accept the electron to form FADH2, right? Then fumarate will be uh, converted into the malate by the hydration reaction, and then malate dehydrogenase will convert malate to oxaloacetate by a oxidation reaction. So malate will be oxidized, so the released electron will be taken by NAD plus to form NADH, right? So one cycle of the citric acid cycle will produce 2 molecules of CO2, 1 molecule of GTP, 3 molecules of NADH, and 1 molecule of FADH2. Right? You can check here also that it will make 3 molecules of NADH, 1 molecule of FADH2, 1 molecule of GTP, and 2 molecules of CO2. One here, right, and one here. So 2 molecules of CO2, 3 molecules of NADH, 1 molecule of FADH2, and 1 molecule of GTP, right? So this GTP is substrate level phosphorylation, right? And this NADH and FADH2, they can release the electron into the electron transfer chain, right? And when the electron will be flown throughout the electron transfer chain in the mitochondria, that it will make ATP by the enzyme called ATP synthase. So they can also, NADH and FADH2, when they will release the electron in the electron transfer chain of mitochondria, in this way they also can produce ATP by ATP synthase, right? That we will see in the our upcoming session, which is oxidative phosphorylation later, right? So these are the product per 1 ton of oxidative citric acid cycle, and then you can see the biosynthetic precursor produced by an incomplete citric acid cycle in anaerobic bacteria. So in anaerobic bacteria, this TCA cycle will be incomplete because there is no oxygen, and you will see that when NADH and FADH2 will release electron to the throughout the electron transfer chain, then the final electron acceptor is oxygen, right? In the electron transfer chain, right? So in this case, if there will be no oxygen, then those electrons released by NADH, so they cannot be accepted by any terminal electron acceptor. So that's why this NADH or FADH2, they cannot be oxidized, right? They will be in the form of NADH or FADH2 if there is no oxygen. So in that case, for example, you can see that in an anaerobic bacteria, when there is no oxygen, then acetyl CoA will make citrate, isocitrate, right? From isocitrate to alpha ketobutyrate, this step will produce one NADH, but there is no oxygen, right? So now this cycle will take a pause here, and alpha glutarate will be used for a biosynthesis of amino acid, nucleotides, heme group, etc. Right? And uh, here, if there is gluconeogenesis in the cell, then this pyruvate will be converted into oxaloacetate, right? So this oxaloacetate in the mitochondria, as we saw in the last class, that this oxaloacetate will be converted into malate. This malate will be oxidized, will be reduced to fumarate, right? So it will, and fumarate will be reduced to succinate, and succinate will be converted into succinyl CoA, just the opposite reaction, right? Then again, the succinyl CoA will be used as a precursor for amino acid, nucleotide, and heme group, right? Because in this case, this succinate to fumarate, it will produce one FADH2, malate to oxaloacetate, it will produce one NADH, but this NADH or FADH2, they will not be oxidized, right? They cannot release their electron in the electron transfer chain, right? Because there is no oxygen. So that's why this NADH level will be high, FADH2 level will be high. So this NADH will be used for this backward reaction or the reverse reaction. So where in the reverse reaction, oxaloacetate will be converted into malate, right? Because NADH level is high. Again, malate to fumarate will be happen. Then from fumarate to succinate will be fumarate will be converted to the succinate, right? Because FADH2 level is high, right? So opposite reaction will be there. So fumarate will be formed to succinate, and succinate will be converted into succinyl CoA, which will be used for the biosynthetic products, right? So in this case, when there is anaerobic condition, then NADH and FADH2, their level will be high because they cannot be oxidized to NAD plus and FAD plus. So they will be used to reduce this oxaloacetate to malate and then fumarate to succinate, right? Just opposite reaction, so that it will form this succinyl CoA, right? Same here, this alpha ketoglutarate, right? As the NADH level is high and NAD plus level is low, so NAD plus cannot be used for this reaction, alpha ketoglutarate to succinyl CoA. So this reaction will be stopped because NADH level is high, NAD plus level is low, right? So this forward reaction cannot be occurred. So that's why this high NADH level, because of this high NADH level and low NAD plus level, this reaction will take a pause, and this alpha ketoglutarate will be used as a precursor for this biomolecules, amino acid and nucleotides, heme, etc. Right? So these are the possible fates of the citric acid intermediates when there is an anaerobic condition. Right? So we will start the regulation. Before that, you can just go through what we covered, and then you can ask me if you have any doubt. So next we will see the regulation of TCA cycle. How the TCA cycles are regulated. As you know that in any metabolism for cells, that has to be strictly regulated. We saw the regulation of glycolysis and gluconeogenesis in the last class. So we will see the regulation of TCA cycle. So from pyruvate to acetyl-CoA, this uh, reaction is catabolized by pyruvate dehydrogenase complex (PDC), and this reaction is depend on the ATP AMP ratio, NADH NAD plus ratio, acetyl-CoA CoA ratio. So if the ATP AMP ratio is high, right, or if ATP is high and AMP is low, right, then this reaction will be prevented. Similarly, if acetyl CoA by CoA ratio is high, then this reaction will be inhibited, right? If NADH and NAD plus ratio is high, then this reaction is inhibited because high ATP means, which is a downstream product. So high ATP means the cell is in a highly energetic state, so there is no need of production of ATP. So that's why this step will be prevented, right? And if there is high amount of acetyl CoA, which is a downstream product, then also there is no need of this reaction. So this enzyme will be inhibited, right? If there is high level of NADH, which is again a downstream product, NADH, which is a byproduct of TCA cycle, as we know. So here also there is no need of if the NADH level is high, that means there is no need of TCA cycle here. So that's why this enzyme complex will be inhibited, right? Similarly, if there is high level of fatty acid, then also it will negatively regulate. But high level of AMP, high level of NAD plus, high level of coenzyme, high level of calcium, which is just opposite of this ATP, acetyl CoA, and NADH. So, right. So, if their level is high, then this step is then this reaction will be positively regulated or activated, right? Because if there is low AMP means, if there is high AMP means low ATP, so cell needs to produce ATP. So that's why it will be activated. If there is high NAD plus, that means there is low NADH. So if there is low NADH, then you need to do the TCA cycle process so that it can produce NADH and it can replenish the NADH pool, right? And if there is high level of CoA, that means there is low level of acetyl CoA. So high level of CoA means there is low level of acetyl CoA. So, right, so this, which is a downstream product, so that's why it will positively regulate the pyruvate dehydrogenase complex. And in other step, similarly, if if high level of ATP, NADH, and succinyl CoA and citrate, both of them are downstream product, right? Succinyl CoA and citrate are the downstream product. So if their level is high, then it will inhibit this citrate synthase, right? And high level of ATP means low level of ATP. If there is low level of ATP, that means cell needs to do the TCA cycle, so it will activate the citrate synthase. For isocitrate dehydrogenase, high level of ATP will inhibit because high level of ATP means, which is a downstream product, so that means cell in a highly energetic state, so there is no need to produce ATP, so that's why the TCA cycle will be slowed down. And if there is high level of ADP, that means low level of ATP. So then cell needs to produce ATP, so it will activate the isocitrate dehydrogenase. So that TCA cycle will be activated, so it can produce the ATP, right? And then alpha ketoglutarate dehydrogenase will be inhibited by the product succinyl CoA, right? And high level of NADH, of course, by the similar reason, and Ca2 plus will activate this enzyme complex, right? So that's how. Those are the regulator of the TCA cycle enzyme, right? So that the TCA cycle is properly regulated. So now we will. So that's all for the TCA cycle. Then if we shift to the glyoxylate cycle, then so in this case, what actually happens, that acetyl CoA, as we saw in the TCA cycle, oxaloacetate combined with acetyl CoA to make the citrate, right? Then by citrate synthase, then aconitase come and it will isomerize citrate to isocitrate. After that, this isocitrate in the TCA cycle, isocitrate would go for oxidation to form alpha ketoglutarate. But in this case, isocitrate lyase will come and it will cleave the isocitrate. Right, right. So it will cleave here. So if I will show you, it will cleave just here, this bond. So it will become this C double bond O minus CH2CH, CH2CH2, C double bond O minus, which is succinate, and it will produce the remaining this portion, which is C double bond O and then C double bond O and CHO, which is glyoxylate, right? So this will cleave this molecule to make 4 carbon succinate and 2 carbon glyoxylate, right? Now this glyoxylate will combine with acetyl CoA to make malate. This acetyl group of this acetyl CoA will attached to here, right, in this carbon, this CH2 and this C double bond O. So it will come and it will attach to this carbon, right? This hydrogen will be removed. So yeah, so what will happen? This C double bond O minus, then CHOH here, and then this acetyl group will come, CH2 C double bond O minus, which is malate, right? That is when the malate synthase will come, and then malate dehydrogenase will oxidize the malate, right? So that this CHOH, this hydroxyl group will be converted into ketone group, right? So if it will be oxidized, then the CHOH will be converted into the ketone group, right? You can see then it will be C double bond minus, C double bond O, CH2 and C double O minus, which is oxaloacetate, which will produce one molecule of NADH, because NAD plus in this oxidation reaction, NAD plus will receive the electron to be converted into NADH, right? And this malate will be oxidized to oxaloacetate. Again, oxaloacetate will attach with acetyl CoA to make citrate, right? So this is occurring in the germinating seeds, where they can use, and I think you guys know that acetyl CoA is a end product of fatty acid beta oxidation. So from right? So from fatty acid, so that's why fatty acid can be used to, in this glycolytic cycle in the germinating seed. So they use their fatty acid. They break down the fatty acid to form acetyl CoA. This acetyl CoA will be used as a precursor for glyoxylate cycle, where they will make glyoxylate, right? And they will release NADH. This NADH, throughout the oxidative phosphorylation, it can produce ATP, right? So that's how they can get energy by ATP generated from this NADH, right? So in the process of glyoxylate cycle. So fatty acid do acetyl CoA, and acetyl CoA will enter into the glyoxylate cycle to make NADH, which will give you ATP, right, for the germinating seeds. So in this case, isocitrate lies in the enzyme, which is not absent in us. So that's why we cannot do the glyoxylate cycle, but it is present in the germinating seeds, so that they can actually use this enzyme to cleave isocitrate to make succinate and glyoxylate, right? Subsequently, it will produce NADH, right? So that's how this is different from the TCA cycle, and they can produce energy from fatty acid. From fatty acid, because the seed can store the fats, so from fats they produce fatty acid, and by fatty acid oxidation they will produce acetyl CoA, which will enter into the glyoxylate cycle to make NADH, which is which will in turn produce ATP by oxidative phosphorylation. So this is the relationship between the glyoxylate cycle and the TCA cycle. So for the glyoxylate cycle in the germinating seed, the lipid body which is stored in the seed, lipid or fat, they will form they are stored as a form of triacylglycerol or TAG. they will be broken down to fatty acid. Fatty acid will be oxidized to form acetyl CoA. Right? This acetyl CoA will enter into the glycolate cycle to form the NADH here from malate to oxaloacetate, and this NADH can be used to form ATP. So here this entire glycolate cycle is occurring in the glyoxysome or the seeds, right? And TCA cycle occurs in the mitochondria. So how they are connected? This isocitrate, there are two possible fates of isocitrate, right? In the glyoxysome, isocitrate can be converted into glyoxylate by using the enzyme isocitrate lyase, right, which is only glyoxysome specific. Alternatively, this isocitrate can be converted into succinate. Succinate can come out from the glyoxysome. It can enter into the mitochondria, where it can participate in the TCA cycle, where the succinate will be converted into fumarate, and then from fumarate to malate, right, and from malate to oxaloacetate. So this succinate can actually feed the TCA cycle in the mitochondria. So two fate, one is participating in the glyoxylate cycle, another fate is participating in the TCA cycle in mitochondria. Right? So that's how TCA glyoxylate cycle and TCA cycle are related in a germinating seed, right? And this malate, which is produced as a byproduct of TCA cycle in the mitochondria, malate can come out from mitochondria to cytosol, where it will be converted into the oxaloacetate by malate dehydrogenase. Then this oxaloacetate can be used for gluconeogenesis, right? So this oxaloacetate, as we saw in the last session, that oxaloacetate can be phosphorylated by PEP carboxykinase to make PEP, and then this PEP will be used for the gluconeogenesis to form the hexose, right? For example, glucose, you can say, right? So that's how this here also this glyoxylate cycle, this isocitrate and enter into this cycle, and it will produce NADH, which will in aerobic condition, which will in turn produce ATP, right? Otherwise, this isocitrate will convert into succinate. It will participate into the TCA cycle. This malate, which is a TCA cycle intermediate, that will come out from the mitochondria to cytosol, and which can take part in the gluconeogenesis to form glucose, right? So it can produce energy, and it can also produce carbon source glucose from this glyoxylate cycle, which started from the lipid body, right? From the lipid body. That's why this lipid stored fat in the seed. That's how the stored fat in the seed can be used to produce ATP here. Also, it can also be used to produce the carbon source, hexose, glucose, sucrose, right? So that's how they are related. So after that, we will move to lipid and fatty acid metabolism. So here we will see how the processing of dietary lipid takes place in the vertebrates. So when we take fat, fat in rich diet, then this fat ingested in the here in the small intestine. So in the small intestine, bile salts emulsify the dietary fats in the small intestine. So it will form mixed micelle. Right? We talked about micelle in the very first class. That micelle is when the fatty acid, which is amphiphilic molecule, which has a both hydrophilic and hydrophobic rich part. So they will assemble and take a shape which is called micelle. So now this intestinal lipase degrades the triacyl glycerol. Right? Degradation of triacyl glycerol will form the individual fatty acid. Right? If you remember from the lipid session, we saw that the lipid or fats are the derivative of triacyl glycerol. Right? There are 2 or 3 fatty acyl group are attached to the glycerol backbone. Right? That's why they are called triacyl glycerol. So if you break down, then you will get individual fatty acids. So these TAGs will be converted into the fatty acid by intestinal lipase. So now this fatty acid and other breakdown products are taken by the intestinal mucosa and converted into triacyl glycerol again. Right? These fatty acids will be assembled to form triacyl glycerol again. Now these triacyl glycerols are incorporated with cholesterol and apolipoprotein into chylomicron. So it will form chylomicron, which is composed of cholesterol, right, and apolipoprotein. Apolipoproteins are the protein which can bind with the lipid, right? So it can bind with the triacyl glycerol, right? So they will make a chylomicron. This green portion is the apolipoprotein, right? And this uh, blue portion is the combination of triacyl glycerols and cholesterol. Right? Now this chylomicron can move in the bloodstream, so it will move through the lymphatic system, right, and bloodstream to tissues. In the tissue, lipoprotein lipase activated by the ApoC2 in the capillary convert the triacyl glycerol of this chylomicron to fatty acid and glycerol. Right? So now it will break down the TAGs to fatty acid, right? And this fatty acid can enter into the cells. TAG cannot enter into the cell. It has to be broken down by lipoprotein lipase in the tissues, right? Then this fatty acid breakdowns can enter into the cells. Here, these fatty acids will be used for the oxidation, so that they can produce CO2 and acetyl CoA, and this acetyl CoA will enter into the TCA cycle, which will create again a CO2 and ATP, right? So this fatty acid oxidation will give you CO2 and ATP, which is where ATP is the source of the energy of a cell. Otherwise, these fatty acids are combined again to make the triacyl glycerol, right? So there are two fats in the adipose tissues. In the adipose tissues, these fatty acids will be combined to make triacyl glycerol and stored as a fat, right? And in the muscle tissues, these fatty acids will be used for the oxidation, so that the cell can produce energy in terms of ATP. For example, we saw here in the amniotic seed, this triacyl glycerol, which was stored, that was used to break it down to fatty acid and then to give you ATP and this uh, exhaust formation, right? So that's how here also in the adipose tissue also we will see that in the adipose tissue also this stored fat can be used for the can be used as a fuel when the cell will need, that we will see, right? But in the skeletal muscle tissue, these fatty acids will be oxidized to form CO2 and ATP, right? So that's how the dietary lipids are processed in the vertebrates. So now we saw the chylomicron, where when the intestinal lipase, when they will degrade the TAGs to fatty acid, then this fatty acid will again taken up by the mucosa and they will be again converted into the TAGs. The TAGs will be incorporated into cholesterol and apolipoprotein, right, which is chylomicron. So this is the structure of the chylomicron. There are three apolipoproteins: B48, C3, C2, right, and uh, this is and apart from the apolipoproteins, there are cholesterol molecules and there are phospholipids, right? Phospholipids are form this spherical chylomicron, and also it is composed of triacyl glycerol and cholesterol ester. Triacyl glycerol is formed by the dietary lipid, right? So now you can see that this triacyl glycerol are packaged to in the form of this chylomicron, which can be uh, traveled throughout the circular system or lymphatic system to different tissues. There, these TAGs will be degraded to fatty acid, and this fatty acid will enter into the individual cells. Right. So now the mobilization of the TAG stored in the antibiotic tissue, as we saw here, that I think the chylomicron is clear because it is made up of apolipoprotein, cholesterol, and the triacylglycerol. This is the molecular structure. There are three apolipoproteins. There are phospholipids. There are cholesterol, and there are triacylglycerol. So this triacylglycerol, this yellow portion, these are packaged to this chylomicron. So the purpose is so that it can travel, right? In the bloodstream is aqueous, right? But your triacylglycerol, they have aliphatic fatty acyl chain, so that is hydrophobic. So it cannot be soluble in the aqueous blood flow, right? So so for that they have to be arranged in such a way. Suppose, yeah, for example here they have to be arranged in such a way so that the hydrophobic portion will be buried inside, and the hydrophilic phosphate group will be exposed to the water, right? So that's how they will make a this is core, right? Hydrophobic core, right? And this phospholipid head will be facing into the outer environment. So that's why they will make a spherical shape where the hydrophobic cores are buried, but hydrophilic heads are exposed to the water. So now they are soluble. The shape is soluble, and the triacylglycerols are packaged here, right? So now they can travel through the bloodstream and lymphatic system, and they can reach into the different cells and different tissues, right? So that is the purpose to deliver the. It is just just a delivery system, so that the triacylglycerol, which are insoluble in water, they can be delivered from the intestine to different tissues of your body. So that's why this is the purpose of making this chylomicron shape. So now we saw that when in the intestine, these TAGs are packaged into the chylomicron, and they will be traveled through the lymphatic and bloodstream system. And when it will reach to the corresponding tissues, then the tissue lipoprotein lipase will break down this uh, triacylglycerol of this chylomicron to fatty acid. So now this free fatty acid, now they can enter into the cell. So if they enter into the myocyte, then the myocyte, they will be used for oxidation to produce energy, ATP and CO2. But if the tissue is adipose tissue, then this free fatty acid will be assembled to form triacylglycerol, right, in a form of triacylglycerol, so that it can be used to produce energy when there is deficiency in the blood glucose level. That we will see in the next slide. So here, how this mobilization of the TAG is stored in the adipose tissue. So in the adipose tissues, the free fatty acids will be assembled to form TAG and will be stored. But when there is blood glucose level is low, then they will be used, right? How? If the blood glucose level is low, then which hormone will be secreted? Glucagon. Glucagon will be received by the receptor in the adipose tissue. It will initiate the signaling cascade, which will eventually activate the adenylyl cyclase, which will make ATP to CMP. Cyclic AMP will activate the CMP dependent protein kinase A. Now this PK will phosphorylate the peripelin, right, which is the boundary of this where the lipid molecules are stored in a form of triacylglycerol, right? So whenever this pKa will phosphorylate this peripelin, so now there will be a hole, right, and this triacylglycerols can come to the cytosol of the adipocyte, right, because of this phosphorylation. And then this pK also can phosphorylate the hormone sensitive lipase to activate it. And this hormone sensitive lipase, when it gets activated by pK, it will break down this TAG to free fatty acids, right? Now this free fatty acid will enter into the bloodstream from the cell. It will export it from the adipose tissue and it will enter into the bloodstream where it will now again free fatty acid is a so it is having a aliphatic long hydrophobic chain, right? So that's why it will be not soluble in the bloodstream. That's why the serum albumin will come. It will bind to the free fatty acid. Now the serum albumin is soluble, so it will just so the fatty acid will be packaged into the serum albumin so that it can travel throughout the bloodstream, right, with the aid of serum albumin, right, and then it will reach to the tissue where it is needed for energy. For example, myocyte muscle tissues, right, in the myocytes or right. So in the myocyte it will be reached and these fatty acid transporters will uh, take out this fatty acid from bloodstream to the myocyte cell where this fatty acid will be used for the beta oxidation to make acetyl CoA and the acetyl CoA will enter into the citric acid cycle which will produce ATP and CO2, right? That means in terms of ATP it will produce energy. So when the blood glucose level is low, that's how then this stored lipid or stored fat which is stored in the form of TAG, they will come, they will actually broken down to free fatty acid which will delivered to the myocyte where it will be used for the oxidation which will produce oxidation followed by TCA cycle which will produce ATP which can provide energy to the cell, right? So that's how in the absence of uh, glucose molecule also, so this is a energy process which is independent of glucose molecule. That's how we can get energy from our from the fat which is stored in our adipose tissue. Yeah. So yeah. So here we can see that that this along with the fatty acid, the triacyl after triacyl glycerol breakdown, it will make fatty acid and glycerol. So what is the role of glycerol? So glycerol by the enzyme glycerol kinase, it will phosphorylate the glycerol at the C3 position, which will produce L-glycerol 3 phosphate. Then it will be oxidized by glycerol 3 phosphate dehydrogenase, right? So that this CHOH will become C double bond O, which is dihydroxyacetone phosphate, which is a glycolytic intermediate, as you know, and that can be isomerized to glyceraldehyde 3 phosphate. We talked about that in the glycolysis, right? So now this glyceraldehyde 3 phosphate can enter into the glycolysis, specifically to the payoff phase of the glycolysis, where it can uh, I mean it can produce NADH, it can produce ATP, right, through this process glycolysis, right? So that's why the glycerol can enter into the glycolytic pathway, which is produced from when the triacyl glycerols are broken down to fatty acid and glycerol. So both fatty acid and glycerol, both of them has role in producing energy, right? So now, yeah. So now, for especially for the myocytes or muscle tissues, so they will use the fatty acid as a energy source to mediate the beta oxidation. For example, here, uh, yeah, here these fatty acids in the myocyte, they will be used for the beta oxidation, right? So this beta oxidation is taken place in the mitochondria. So now for that, this fatty acid has to be transported from the cytosol to mitochondria, right? So how can it be transported? So that process is called Carnitine shuttle. So here basically in the cytosol there is acetyl CoA, right? Yeah. So in the cytosol, this fatty acyl group will be attached with a coenzyme group, right? So that it will make a uh, fatty acyl CoA, right? Now this fatty acyl CoA will combine with carnitine to release this coenzyme A, and it will become coenzyme A will be replaced by the carnitine, right? So this will become RC double bond O carnitine, right? So this is called carnitine acetyl. Yeah, so it is called the fatty acyl carnitine. Now this fatty acyl carnitine is a substrate for carnitine acyl transferase 1. So it will, uh, yeah, so it will transfer this fatty acyl carnitine to the from the cytosol to intermembrane space uh, of the mitochondria, and in the inner membrane there is a transporter is there. So throughout this transporter, this fatty acid carnitine will enter into the matrix of the mitochondria, right? So now this carnitine group, the role of carnitine is over. So now the carnitine acyl transferase 2 will replace the carnitine, right, with the coenzyme A. So this carnitine will be eliminated and coenzyme A will bind to this fatty acyl group. So it will become carnitine will be removed and it will become again fatty acyl CoA, right? That is mediated by the carnitine acyl transferase 2, right? So now this fatty acyl CoA can in the matrix, in the mitochondrial matrix, this fatty acyl CoA will get access of the TCA sorry, so it will get access of the beta oxidation enzyme and it can enter into the beta oxidation. So these are the stages of the beta oxidation. So we will first start with the unsaturated fatty acid, which is palmitoyl CoA. So here first step is FAD plus will be used as a cofactor. It will be reduced to FADH2. So that means there is oxidation process, right? So here there will be double bond between alpha and beta carbon, right? So there is a double bond between alpha and beta carbon, which is catalyzed by acyl CoA dehydrogenase. Now enoyl CoA hydrogenase will come, so it will add an molecule of water, right? So here an OH molecule, here one H molecule. So it is CHOH, CH2, right? So S coenzyme A is C double bond O, CH2, CHOH, right? So this is beta hydroxyacyl CoA, right? After that again a step of oxidation which is catalyzed by beta hydroxyacyl CoA dehydrogenase. So where this OH group will be oxidized to C double bond O, ketone 2, right? So it will become beta, because at the beta carbon is oxidized, so it is beta ketoacyl CoA. Next acyl CoA acetyl transferase will come, or thiolase. It will add one more molecule coenzyme A. So now it will become So now it will split it, right? So the split will be here actually. This is alpha carbon, this is beta carbon. So the split will be here, right? So it will be form one molecule of this CH3C double bond O SCOA, which is acetyl CoA, and one molecule of RCH3CO SCOA, right? So initially it was C16. Now one acetyl CoA is removed, that means which is a 2 carbon molecule. So C16 minus C2 is C14. So it is remaining of C14. Again this C14 will again it will enter into the beta oxidation. Then after one round of beta oxidation, it will make one molecule of acetyl CoA and it will become C12. Then again C12 will make one molecule of acetyl CoA C10. C10 will make one molecule of acetyl CoA C8. C8 will make one molecule of acetyl CoA to C6. C6 will make one molecule of acetyl CoA to C4. C4 will make two molecules of acetyl CoA. So total 1, 2, 3, 4, 5, 6, 7, 8 molecules of acetyl CoA will be produced, right? And so total 8 molecules of acetyl CoA will be produced when after complete oxidation of this C16 palmitoyl CoA, right? So each of the time these reactions will be repeated, this cycle will be repeated, so that and after complete oxidation it will make 8 molecules of acetyl CoA. Each of them will enter into the TCA cycle to produce energy, right? So that's how fatty acid can give you energy by beta oxidation. Here you can see a summary. that here after stage one, so after full oxidation, it will make 8 molecules of acetyl CoA that will enter into the TCA cycle to make 16 molecules of CO2, right, and 64 molecules of electron by in the form of NADH and FADH2, right, which can actually transfer the electron to the electron transfer chain to make ATP. Right, so that's how this fatty acid beta oxidation will produce acetyl CoA that can actually produce CO2 and ATP. Right, so this is the stage. So these are the stages of fatty acid oxidation. So that is the balance sheet for one round of beta oxidation from the palmitoyl CoA. So it is consuming one molecule of NAD plus, one molecule of FAD, right, and one molecule of coenzyme. See, it is one molecule of FAD, NAD plus, and coenzyme, and of course one molecule of H2O, because in this step H2O is used, right? So it is forming the myristoyl CoA, which is C14, which is a C14, right? And then it is forming one molecule of acetyl CoA, of course, and then it is producing FADH2 and NADH, one molecule, right? FADH2 and NADH, one molecule, right? After full oxidation, it will use, there will be 7th cycle. So 7th cycle will use 7 coenzyme, 7 FAD plus, 7 NAD plus, 7 molecules of H2O, and ultimately it will produce 8 molecules of acetyl CoA and 7 FADH2, 7 NADH, right? So that is the after full oxidation, that will be the balance sheet for the beta oxidation of fatty acid for C16 palmitoyl CoA. So now if you will see the oxidation for unsaturated fatty acid, so if you will see the oxidation of the unsaturated fatty acid, see, suppose this is the oleoyl CoA, which is C18, and there is a double bond here, unsaturation. So then after 3 cycle of this beta oxidation, as we saw, after beta, so the cleavage is done between alpha and beta carbon. This is alpha and beta, so this is the region of cleavage. So here also, this is the alpha and beta carbon, so the cleavage will be there. After 3 cycles, you will get 3 molecules of acetyl CoA, and you will get C12, which is dodecanoyl CoA, right? Now there is a, if we want a cleavage here, alpha and beta, then there is an unsaturation, so cleavage cannot be happen. So in this case, there will be a isomerization between, which is catalyzed by enoyl CoA isomerase. So this double bond will come here, right? So now, so now the cleavage should be in the between alpha and beta here, but there was a saturation, so it was not possible. So it shifted to between alpha and beta. So now there will be cleavage between alpha and beta. So it will produce one molecule of So it will produce. So when the cleavage is here, then so then it will produce one molecule of acetyl CoA and C10, right? So after that, so after that, next cleavage will be here. Next cleavage will be here. Now there is no unsaturation, and next cleavage is here. After this cleavage, cell will produce 2 molecules of acetyl CoA. So total, after this 3, 4, 5 beta oxidation from here, it will produce 5 plus 1, 6 acetyl CoA. If there is n number of beta oxidation, it will produce n plus 1 number of acetyl CoA, right, after full oxidation. So that's why, so that's how the unsaturation will be tackled, right? So it will produce total 6 molecules of acetyl CoA. Now here, if there is mono unsaturation, right, so This was the mono unsaturation, but if there is more than one unsaturation, then how the cell will take place? So here it was oleic acid where one unsaturation, but in case of linoleic acid, the C18, there is two unsaturation, C9, C10 and C12, C13. So first this alpha beta cleavage will be there. So 1, 2, 3. Now in the next step, after 3 round, there will be 3 acetyl CoA, right? And it will the length will be C12. 3 into 2 6, so 18 minus 6 is 12. C12. So now after this cleavage, the cell will produce uh, yeah. So this molecule, right? So now if we want to cleave at alpha and beta, right, then there will be a vicinity double bond. So not possible. So again delta 2 delta 3 NOL CoA isomerase will come. It will shift the double bond to here between alpha and beta. So again this cleavage will be there, right? So after this round, one cycle of beta oxidation, the cell will produce C10 and one molecule of acetyl CoA. Fine, fine. Now after this, yeah. So after this, you can see that there will be yeah. So this C10 molecule will be there, and yeah. So and there will be a fast oxidation of the second cycle. So it will make a double bond between alpha and beta, right? So double bond is he, so alpha beta is here. So here a double bond will be placed, right? After this one cycle. So now there is two double bond, right? So now this 2,4 di enoyl CoA isomerase CoA reductase will come, so it will shift the double bond to here, single double bond, right? So it will actually reduction, which will oxidize the NADPH to NADP plus. So now the double bond will plus here, will be placed here. So next, yeah. So now the cleavage should be there, but again there is a double bond. So that's why again the same thing like happened here. Double bond will be shifted to here, right? Shifted to here. So now this alpha beta cleavage will be there, right? So right, so which will after this step it will produce C8 and another one molecule of acetyl CoA, and subsequently 1, 2, 3, 4 cleavage will be there, right? 4 cycles will be there. So it will make from here, it will make after 4 cycle you will get 5 acetyl CoA for full oxidation, right? So here there was total cycle was 3 here, 3 here, 1 here, 4, right, and 5 here. Sorry, 3 here, 1 here to 4, and then again 4 here to 8 cycle. So after 8 cycle you got 3 acetyl CoA here, 1 acetyl CoA here, 4, and then 5 acetyl CoA here. So total 9 acetyl CoA. So 9 acetyl CoA you got, right? 9 acetyl CoA and number of cycle total was 8, right? 8 cycle. Right? See, the rule is followed. If the number of cycle is n, the number of acetyl CoA you will get is 8 plus 1, 9. So in this case, when there is mono, so when there is polyunsaturation, the cell will tackle in this way, right? So yeah, so that is the oxidation of the unsaturated fatty acids. And now we will see the coordinated, yeah. So now we will see the coordinated regulation of the fatty acid and synthesis. Because in the cell, fatty acid oxidation takes place in the mitochondria, and also fatty acid synthesis is also required. So how this regulation between the fatty acid breakdown and synthesis will be happening. So when so when the blood glucose level is high, when you are getting a carbohydrate rich meal, your blood glucose level is high, then then your body will secrete insulin hormone, right? This insulin hormone will activate one phosphatase which will remove phosphate group from this acyl CoA carboxylase, right, which is a enzyme for fatty acid biosynthesis. So it will remove the phosphate, right, so that it will make it active. Phosphate form is inactive, so upon releasing the phosphate, it will be active, right? So now this acyl CoA carboxylase, it will use acetyl CoA, right, to make malonyl CoA, right? Now this malonyl CoA can be used uh, to make fatty acid, right? And this malonyl CoA can be converted into fatty acyl CoA, right? This fatty acyl CoA can go inside to the mitochondria by this carnitine shuttle. So that's why this carnitine acyl transferase 1 is required here, right? To make fatty acyl CoA to fatty acyl carnitine, so that it can go to the matrix of the mitochondria, fatty acyl carnitine, which will again transfer the carnitine to make fatty acyl CoA, right? Then it can take place for beta oxidation, right? So malonyl CoA will inhibit this process. This fatty acyl CoA to fatty acyl carnitine, right? For getting inside the mitochondria, fatty acyl CoA has to be converted into fatty acyl carnitine by carnitine acyl transferase 1. Otherwise, it cannot be transported. But this enzyme is inhibited by malonyl CoA, which is a fatty acid biosynthesis enzyme, right? So, if it is inhibited, then fatty acyl CoA cannot be converted into fatty acyl carnitine, so it cannot be entered into the mitochondria. So, there will be no beta oxidation. So, that's why malonyl CoA will negatively regulate the fatty acid beta oxidation. It will inhibit the fatty acid beta oxidation, right? So when this acetyl CoA carboxylase will be activated by the phosphatase, then it will convert the acetyl CoA to malonyl CoA, and this acetyl CoA is coming from the glycolysis, because after getting a dietary carbohydrate rich meal, you will get high amount of glucose. So glucose can be converted into acetyl CoA, right, by the glycolysis followed by pyruvate dehydrogenase complex. So glucose will form acetyl CoA, right? Now this acetyl CoA will be converted to malonyl CoA by acetyl CoA carboxylase, right? So now this malonyl CoA will be converted into fatty acid, which is a step of process of fatty acid biosynthesis, right? So that is the fate of when the blood glucose level is high. When insulin is activated, it will activate the acetyl CoA carboxylase, which will produce malonyl CoA, which will produce fatty acid biosynthesis, right? And also this, as I told you, malonyl CoA will inhibit this carnitine acyl transferase 1, so that it will inhibit the entry of the fatty acyl CoA into the mitochondrial matrix. So it will inhibit the beta oxidation. So high blood glucose actually secrete the insulin, which will activate the fatty acid biosynthesis, and it will inhibit the fatty acid beta oxidation, right? But when there is blood glucose level is low, then glucagon will be secreted, which will activate the protein kinase A. Now this PK will phosphorylate the ACC, acetyl CoA carboxylase, so it will be activated. So now there will be no malonyl CoA, right? So if there is no malonyl CoA, then there will be no fatty acid biosynthesis, right? Because now you need energy, you need beta oxidation when the blood glucose level is low. So then, in the absence of malonyl CoA, the negative regulator is absent. So now this fatty acyl CoA can be converted into fatty acyl carnitine by this carnitine acyl transferase 1, because the regulator is not present, and then this fatty acyl carnitine can go into the mitochondrial matrix, and it can undergo beta oxidation to produce acetyl CoA, means energy, right, which can enter into the TCA cycle and produce energy. So when blood glucose level is low, you need energy from fat. So now this protein kinase A will inactivate acetyl-CoA carboxylase. As a result, there will be no malonyl-CoA, and in the absence of the malonyl-CoA, fatty acid synthesis will be prevented, and malonyl-CoA, and in the absence of the malonyl-CoA, fatty acyl-CoA can be entered into the mitochondrial matrix to initiate beta oxidation. So it will activate the beta oxidation, right? But it will inhibit the fatty acid biosynthesis. So that is the regulation of fatty acid breakdown and the synthesis. When high blood glucose level will inhibit the beta oxidation and will activate the fatty acid synthesis, and low blood glucose level, when you need energy, then fatty acid biosynthesis will be reduced and the fatty acid beta oxidation will be activated to get energy, right? That's how the fatty acid breakdown and the fatty acid synthesis is coordinately regulated. Uh, Apart from mitochondria, in peroxisome also there is beta oxidation, you can see. Uh, so if we just compare between the beta oxidation of peroxisome and mitochondria, so this is the fatty acyl-CoA is here. Then in the first step is similar, that FAD will be used, right, for the oxidation process. So there will be double bond between this alpha and beta, right? And this FAD, right? So for this mitochondria, FAD will be reduced to FADH2, right? And this FADH2 will release the electron to the respiratory chain, right? So that FADH2 will be converted into FAD, right? And the electrons which is released in the electron transfer chain, they will flow through the electron transfer chain and it will be accepted by oxygen to make H2O. And this electron flow throughout the ETC, it will produce ATP by ATP synthase. But for the peroxisome, this when this FAD will be reduced to make FADH2, then FADH2 will not release the electron to the electron transfer chain, right? Rather, it will donate the electron to the oxygen to make H2O2, right? This H2O2 will be broken down to H2O and half O. So there will be no ATP synthesis because the electrons are directly given to oxygen to make H2O2, unlike the mitochondria where the electrons are flown through the electron transfer chain which can produce ATP. So in the mitochondria there is production of ATP, in the peroxisome there is no production of ATP. Then in the next step, H2O will be added here, which here and CH2 here. Then again oxidation step, right? So this OH will be converted into the C double bond O. Here also this in the mitochondria NAD plus will be used for the oxidation. So NAD plus will be reduced to NADH, and NADH will release the electron to the respiratory chain, right? Which will actually produce ATP when this electron will be flown through the electron transfer chain to accepted by O2. So it will reduce oxygen to H2O and it will produce ATP by ATP synthase. But in this case, peroxisome, NADH will be reoxidized to NAD plus, right? So it will not produce ATP here. Then again, the same thing, coenzyme will be added here, right? So and there will be a cleavage between alpha and beta here. So it will get RCOSCO and the CHCCO-SCO, acetyl-CoA, right? So for the mitochondria, acetyl-CoA will enter into the citric acid cycle, but for glyoxysome or peroxisome, the acetyl-CoA will be exported from the peroxisome. So the differences are in first oxidation step of FAD to FADH2. So in the first oxidation step, this FADH2 will release the electron to the electron transfer chain to produce ATP, but here FADH2 will directly donate the electron to oxygen which will make H2O2. So there is no production of ATP. Same here, NADH in the mitochondria, they will also release the electron to the electron transfer chain and it will produce ATP. But in this case, peroxisome, NADH will be reoxidized to NAD+, there is no production of ATP. And this acetyl-CoA, which is the product of fatty acid beta oxidation, in mitochondria it is used for the citric acid cycle. In peroxisome it will be exported, right? So there is no ATP production in the peroxisome, but in the mitochondria, the byproducts are associated with the ATP production, right? And in the seed, TAGs or triacyl glycerol as this is a source of energy. So seed triacyl glycerol which are stored, they will be by using lipases, they will be broken down to fat, and fatty acid, free fatty acid. After beta oxidation, fatty acid will be converted into acetyl CoA, and as we know that it will enter into the glyoxylate cycle. You can see this fatty acid will be enter into the glyoxylate cycle, right, after producing acetyl CoA from beta oxidation. So in glyoxylate cycle it will produce NADH, and also it will produce isocitrate, which will convert into succinate, which will come to mitochondria to take part in the citric acid cycle, which will produce ATP. Right? So same thing is described here. So you can see that TAG are energy source in the seed. So, so lipase will break down to fatty acid. In mitochondria, beta oxidation will produce the acetyl CoA. In glyoxysome, acetyl CoA will enter and it will it will make the oxaloacetate in the glyoxysome, and this oxaloacetate will take part in the gluconeogenesis to make glucose. Right? The glucose is the precursor for sucrose, polysaccharide, amino acid, and also energy, because glucose can enter into the glycolysis and TCA cycle to produce ATP. Right? So that's how TAGR energy source in the seed. If you again see this diagram here, then you can understand properly that how this acetyl CoA is how it is converted, how it is participating in the glyoxylate cycle. So from there succinate is coming. Succinate is coming to TCA cycle, mitochondria. It is forming oxaloacetate. Right, so it is forming actually malate. Malate is exported to the cytoplasm, which will be converted into the oxaloacetate. Right, that's how this acetyl CoA is converted into the oxaloacetate. Now this oxaloacetate is entering into gluconeogenesis to produce glucose. Right, same here is shown here that in the seed, fatty acids are broken down by lipase, and then beta oxidation will produce acetyl CoA. This will enter into the glyoxylate cycle, and it will make oxaloacetate, which will be used for gluconeogenesis, as I told you, described you. So by gluconeogenesis it will produce glucose. Glucose is a precursor for sucrose, other polysaccharides, amino acids, nucleotide, metabolic intermediates, and it will enter into the glycolysis and TCA cycle to produce energy in terms of ATP. Right? So that's why triacyl glycerols can serve as an energy source in the seeds, right? And it can also serve as a glucose source in the seed, right? So that's how seeds can make carbohydrate glucose from fat, from lipid, right? By taking the help of glyoxylate cycle. And in the endoplasmic reticulum, there is another oxidation, which is omega oxidation. So of fatty acid. So here basically the omega position is the terminal position of which is the most distant carbon from the carbonic from this carboxylic acid group. So this mixed function oxidase will oxidize the CH3 group. So it will by using the oxygen, so it will become CH2OH, and then alcohol reagents come and it will take Na plus as a cofactor, and it will oxidize this this OH group to aldehyde group, right? So this is CHO CH2 10 acid COO minus. Again oxidation by alcohol dehydrogenase, which is again used Na plus as a cofactor. So this aldehyde will be converted into acid, right? And the electron is received by Na plus to make NADH. So now it is having CH2 10, and in the two terminus there are two carboxylic acid. So now it will take part into the beta oxidation to make succinate instead of acetyl CoA and adipic acid. Right. So that's how it will make after beta oxidation instead of acetyl CoA, it will make succinate and adipic acid. Right. So these are the end product of the omega oxidation in the endoplasmic reticulum. Right. So yeah, that's how from our session today. Fatty acid catabolism we discussed, and then we also discussed the right, and we also discussed the TCA cycle. One carbohydrate metabolism and one fat metabolism we discussed. So if you have any question, you can ask. Okay. So I think that's all for today's session. I think we can close the session. See you on the next week. Next week. Thank you for joining.